Hey, let's get started. It's a great pleasure to welcome Jens Skotheim to our timeline series. So, yeah, I've known for quite some time. I was just looking up your pedigree. Jan had trained in got undergraduate, did undergraduate math and physics at MIT, and then went on to the real Cambridge to do a PhD in applied mathematics. Not sure what your thesis was, but was it related to biology?
Yeah, some thought matter.
Okay, okay. Because then
Yeah.
I see several papers with you and Amaka sort of after this. That's probably done that.
That's the PhD.
Huh?
He was my advisor.
Oh, he was your advisor.
Yeah.
He was back. Oh, then, okay, okay. I was your mathematical biology shapes and the forms and the math and things. And then, but then you make the transition to Rockefeller to pick up experimental cell biology. And, that's what I first met him in the front class level. He was a postdoc. I looked at this cell cycle and the cell size control. That's and then he's since been at Stanford, or since what? Since beginning of 2010,
2008.
Yeah. Okay. Right, and as you will see, has established himself as a major force in quantitative study of eukaryotic cell biology, cell growth, cell size. So we're look forward to conference.
Yeah. So turn this on. So first of all, Terry, thanks for the invitation to come here, and I've really enjoyed everybody I've met with today, and already learned a lot. So it's really been a pleasure. So today I'm going to talk about really my kind of like favorite question in cell biology, and that's this question of how it is that cell growth triggers cell division. And in fact, the question as posed is really simple. A lot of cells grow, and there's kind of a growth requirement for many different types of cells in order to enter the cell division cycle and divide. And so ultimately, what my group has been working on, what I've been thinking about now for over 15 years, is how is it that this works, right? So what is it that's biochemically different about a large cell compared to a small cell that makes it enter the cell division cycle and divide, right? So it's a very, very simple-minded question. It was the question that brought me to molecular cell biology, in fact. So when I came to the Rockefeller University to work with Eric Sigia and the Center for Studies in Physics and Biology, Fred Cross and his group that kind of like implemented one of these time-lapse microscopes, sort of made popular by the work of Mike Elowitz a couple years earlier, right? So we were looking at these movies of growing and dividing, but these cells are expressing these fluorescent proteins, and this sort of question of, why does it divide when it divides? Clearly it has to grow to divide. How does this happen? This was, a simple enough question that even someone with a physics training could appreciate. Not like these complicated molecular biology questions that most people are involved in. Anyway, so that was the hook that got me into making movies. And so then once it's a slippery slope, right, Terry? Like you start making movies and analyzing them, and then all of a sudden you're, mutating genes and purifying proteins and all kinds of terrible things. But here we are. So this is this question: how do cells link growth to division? I think it's an important question because this is ultimately what allows cells to control their size, to be able to achieve some kind of target cell size, which is really at the core of different types of cells, right? So if you look at different types of cells, what you'll see is like one of the most noticeable differences between these different types of cells are, in fact, they're massive differences in size. And so if you look across the human body, if you look here at a million-fold range in cell sizes, what you'll see are that's linked to function. And so you can quite easily appreciate that at the extremes of the size scale. So you have things like sperm cell and red blood cells, which, you know, obviously need to be small in order to fit through tight spaces and do what they need to do. And then you have kind of like, you know, bigger cells, like biggest mitotically competent cell would be the oocyte. And it's massive, and it really needs to be massive because it has to have all of the material in order to make the, you know, over 100 cells of the pre-implantation embryo, and which then, you know, the pre-implantation embryo can then implant and start getting nutrients from mom and start growing on its own. But before that, this, you know, beautifully large cell has to supply a lot. So it's quite big. Right. So while different types of cells have different characteristic sizes, if you zoom into any one particular type of cell, what you'll see is a group of cells that look quite similar. And what that really means is that cells have a way of kind of like measuring how much they grew and then linking this information to either a division or a differentiation process, right? And so our question was, you know, how does this work from a molecular point of view? When I started thinking about this and introduced to this question in Fred Cross's lab at the Rockefeller, I would say the state of the field was that we didn't know how this worked for any type of cell, much less how a process like this could be, you know, modulated and over, let's say, a million-fold range in target cell sizes of the human cells that I showed you before, right? So we didn't know all that much. And our approach was really, let's try to work with the simplest, you know, most well-behaved eukaryotic model system that we could think of, and that is, of course, the wonderful budding yeast. Okay, so how does it work in budding yeast? Well, it's been known since essentially the beginning of time, when Lee Hartwell roamed the earth, that the G1S transition is this key moment in budding yeast cells where growth is linked to the cell division cycle. And if we crack open the textbook published in 2008, the year I started my lab, this is the textbook on cell cycle control by Dave Morgan from UCSF. What you'll read is that the G1S transition, like the other transitions in the cell cycle, are driven by these changes in activity of cyclin and cyclin-dependent kinase complexes, right? So the question as posed to us at the time was, you know, how do you get some kind of increasing size or growth dependent of increase in this activity, right? And so, you know, I know there are physicists in the audience, and I apologize in advance, but I have to introduce some like 3-letter acronym, you know, protein names. There's no way around it, but I'll try to introduce as few of them as I can for your benefit and enjoyment. Okay, so in budding yeast, what does this mean? It means the cyclin called CLIN3, which forms a complex with the single cyclin-dependent kinase in yeast, CDK1, and then this kinase complex, you know, activates a series of biochemical events that leads you into the cell division cycle. But let's forget about all of that for now. You know, the question that we were really trying to address circa 2008 was how does this happen? How do you get like some size or growth dependent increase in the activity of this complex? And so we did what, you know, we like to do, which is single cell imaging. And so we tagged the gene at the endogenous locus with the fluorescent protein, and then we measure, you know, what happens, right, in time and space, the concentration of this protein. And so we make a bunch of movies. This is just like a typical movie of that we made during my postdoc of some fluorescently, you know, growing and dividing yeast. I think the movies are quite beautiful. And from these kind of movies, we can, you know, segment the cells and track the cells and calculate trajectories of concentration dynamics. And so we did that for CLIN3. I don't want to get into all the details of technical complications there were to do this, but in the end, the result was this, right? So basically, the concentration of the CLIN3 protein does not change very much as the cells grow and get larger in the G1 phase of the cell cycle, right? So in a way, CLIN3 is the cyclin that doesn't cycle. You know, this observation rules out maybe some of the simplest versions of this textbook model. I should also say here that we weren't the first ones to observe this. So Mike Tyers and Bruce Futcher in 1993 saw essentially the same thing, looking at the mRNA of CLIN3 in one of the first papers characterizing the dynamics of CLIN3 mRNA, right? So they, so they took a break from, I don't know, hunting mammoth or whatever you did in 1993 and put away their stone tools and measured the CLIN3 mRNA. But we basically got the same result with our fancy single cell analysis. Right. So then we're stuck with this question, that what's supposed to be the size dependent trigger of the cycle that we're measuring is not really looking much like a trigger, right? So we're stuck with this protein which concentration isn't changing, and we're wondering, how do you generate a kind of size dependent signal out of this concentration which is not changing. And so we thought long and hard about this question, proposed a bunch of different kind of like interesting biochemical models for how this could work. Ruled them out one by one through quantitative measurements over a span of a couple of years, and we're still stuck staring at the same question. And we're basically stuck staring at this question until Kurt Schmoller, who was a postdoc in the lab at the time, asked a very simple yet, deceptively deep question, which was, what about all the other proteins in this pathway? What if we look at their dynamics, right? And so Kurt tagged everything in sight with green fluorescent protein and measured its concentration dynamics. And so you found basically a whole lot of proteins where the concentration wasn't changing very much, except for one protein, right? So this one protein, which was the exception, was this protein, We5, and this got us, unbelievably excited at the time, because what do we see here? We5 is, it was shown in these two publications that it's an inhibitor of cell division cycle. And what Kurt measured is that the concentration is decreasing in inverse proportion to the volume of the cell in the G1 phase of the cell cycle, right? So then this is exciting because okay, it's not that exciting to just throw mud and rule out some models. It's really exciting to try to generate new models that can explain in a more comprehensive way the phenomena that we all observe. And so this was exciting because there's a new model is saying that size and growth could feed into the middle of the pathway. This protein We5, which is an inhibitor of cell division, sitting at the middle of the pathway, and growth quite literally dilutes it out. So I won't go into all the details of experiments, about that, because this is now, getting increasingly old in this paper. But it's still a good paper. It's still true. If you want to read it, it's still, I think, a pretty interesting one. Okay, so we're making some progress on this question of how cell growth triggers division. We introduced a new mechanism, and this mechanism we introduced is what we call inhibitor dilution. So basically, as the cell grows, it gets bigger, and as the cell gets bigger, the concentration of these inhibitors of cell division decrease, and that drives the cell into the cell cycle. Right. So why did it take so long? It's maybe a little bit embarrassing, but we had all the tools there in my postdoc to solve the question, at least half a dozen years earlier. And we didn't, and it wasn't any kind of technical problem. It was really a conceptual problem. So basically, we were thinking cells are getting bigger, they have more different proteins. We were stuck on trying to understand this idea of how do we generate a size-dependent increase in some activity, some activator. And it took us a long time to come around to the idea that, really any protein can be a growth sensor. It doesn't have to be an activator. And in fact, an inhibitor, if you dilute it, would work just as well. All that needs to happen really is that the protein concentration changes with cell size, and then there's an associated activity which is important which changes with that protein concentration. So that was kind of the conceptual innovation that we were missing in order to write that paper or make that progress. So then we wanted to know, well, can we apply this idea in other systems now that we have this new idea? And so we thought, why not look at the human cell cycle, which there are a lot of aspects of it that are conserved. You have cyclins and cyclin-dependent kinases that are sort of similar types of proteins. You have a bit more inhibitors of the cell cycle that appear here and there that don't really exist. So that's the thing with human cells. They just have an annoying number of genes. Yeast's much better, much much fewer numbers of genes. It's easier to deal with. But anyway, so you have kind of like more inhibitors here or there that could possibly be playing a role. Ideally, what would we do? Well, ideally, we wanted to do, and this was around the time where, CRISPR was so big and just like exploding everywhere. We're like, let's use CRISPR to tag everything and look at dynamics just like we can in yeast. It turns out that was really hard. And after a lot of effort, we ended up tagging basically 2 genes at their endogenous loci, these famous cell cycle inhibitors, P21 and the retinoblastoma RB. And so the question was, are these in any way sensors of growth like the inhibitors in yeast were? And so we looked at that, and you can appreciate that that's not true for P21, so not so true for P21. And we weren't the first person to observe the P21 dynamics like that. There's also Alexis Barr's group in London observed the same thing. But what about RB? So we can look at RB, and in fact, the dynamics of the retinoblastoma protein look a whole lot like what we found for WE5, and that the concentration of RB is just decreasing as the cell is going through the G1 phase of the cell division cycle. So very, very similar story. This is okay. Right. So here we are with this issue of how to generate a link between growth and cell division. And we identified this inhibitor dilution model, first finding that the protein WE5 in yeast is being diluted, and then later finding that the retinoblastoma protein in human cells is also being diluted in the G1 phase of the cell cycle. And since our work was published, there's 2 further reports. There's quite a nice paper in looking at Arabidopsis plants, where they found a totally different type of cell cycle inhibitor, KRP4, which is diluted in G1 to achieve a similar aim. And then there's also a paper in looking at Chlamydomonas alga, finding this protein tiny1 is also diluted out in a similar way. Yeah.
The concentration you're referring to is a nuclear concentration?
Yeah. I mean, we typically measure cellular concentration. All of these proteins are in the nucleus, basically exclusively, but the nuclear volume really stays in the condition that we're looking at, really stays as a constant fraction of the cell volume. So they're equivalent statements. So the nuclear concentration is doing
G1 when the chromosome is not increasing, the nucleus size is still increasing.
Yes. And the nuclear size is really independent of the number of copies of the genome and determined exclusively by the cell size we've seen. So their experiments, best experiments are in fission yeasts, where you can generate these kind of like big yeast cells that either have 1 or 8 copies of the genome. And for the equivalent size yeast, the nuclei are exactly the same volume. And it's always about 10% of the cell volume.
Is that correlation?
Yeah, that correlation is absolutely beautiful. It's done by some Swiss people, but very nice. Okay, so that was this was our kind of big result. And so going from that result, like over the last 5 years, that's really generated, I think, 4 new and important directions that we're currently working on. So one direction that we've been working on is to try to understand what's the general pattern of synthesis across the proteome. So what we saw was, or I don't know how to, as cells grow and get larger, typically the concentration of total amount of proteins in the cell stays roughly the same. So our default thought was, well, probably most proteins stay at constant concentration, and then there's some interesting outliers like WE5 and RB and histones, which are being diluted as the cells grow. But then most proteins would stay around constant concentration. That's not what we observed. What we observed is actually that there's, even though the total protein concentration stays the same, there's really extensive and very interesting remodeling of the proteome as the cells are growing larger and larger. We now have published quite a few papers on this. Secondly, of course, we're interested in, WE5 and RB have these really interesting and specific dynamics that are important for a cell division cycle. So we want to understand, molecularly, where does that come from? Third, I would say that now that we've been thinking about, how do we understand the molecular mechanisms giving rise to these diluted proteins, it's made us rethink and kind of think back on how do you even generate a single protein which stays at approximately constant concentration as cell grows? That's also not, the more you think about it, the more you realize that this isn't a trivial problem either. And so we made some progress on that in the paper we published late last year. And then finally, and this is to to try to address some, like, I guess, deep personal fears that I have working on tissue culture cells of human and mouse tissue culture cells, growing on glass dishes, plastic dishes, is, what does all this really mean? Like, are these kind of physiological questions of size control? I mean, is this even a relevant model? Does this make sense? Like, or, is this work in cell culture like very much an artifact? And so we've been trying to come to grips with that by doing some quite technical intravitreal imaging of cells growing within live mice, which I'll tell you about on the second half of the talk. So the first half of the talk, I'm going to talk about what we've learned about the regulation of RB protein concentration dynamics. And then in the second half, I'm going to talk about this, try to connect with the real physiology of this cell. So this is for you, Terry. Okay, so much like the yeast pathway, there's similar positive feedback loops in the mammalian G1 control. And so here we're talking about the central protein, the retinoblastoma protein, which we found was diluted out by cell growth. In addition to the dilution by growth, it's an important target of the upstream cyclin D CDK4 kinase. It phosphorylates the retinoblastoma protein. And then it can be RB can be completely phosphorylated by downstream cyclin E CDK2 complexes. So it's RB is not only regulated by the concentration, it's also regulated by the phosphorylation. And in fact, this phosphoregulation of RB is very much the canonical way we think about RB concentration. In fact, there's a cancer drug which targets this kinase, which just sold for about, they sold $20 billion worth of that cancer drug. And that's targeting the ATP pocket of this kinase to stop it from phosphorylating RB. So this is very much the dominant way of thinking about RB inactivation is phosphorylation. We introduced, this RB concentration pathway, which is sort of an independent pathway to inactivate RB, where, as I showed you earlier, the concentration decreases through G1 phase of the cell cycle. This concentration reflects the cell size, right? So here we're looking at the size of the cell at birth versus RB concentration of that cell, and you see the nice inverse correlation there. And the concentration of RB is important for the proliferation, right? So if we measure the rate at which cells transit across the G1S transition, the rate at which cells go through the G1S transition as a function of the RB concentration, you can see that the higher the RB concentration, the lower that rate of transition is. So what do we have here? We have like kind of two pathways of RB inactivation. If we look at the concentration of RB through the cell cycle, it's kind of decreasing in G1 and then goes up again in SG2 phase, where it's inactive because it's phosphorylated. All right. So where does this dynamics come from? Well, in principle, it could be any part of this process, right, leading to protein synthesis and degradation. Any one of these steps could have some size dependence or some cell cycle dependence that would give rise to the observed dynamics. And so we, tested all of these aspects of synthesis. I'm not going to show you the data because it's all basically negative results. And that left protein degradation. And so let's look at that. So I'll show you the kind of key experiment which convinced us that protein degradation was the answer here. And so what we do is we introduce into these human cells, we introduce the doxycycline-inducible allele, which expresses RB fused to a green fluorescent protein. We can wash out the doxycycline, and then we can observe how the total protein intensity, the amount of that protein, decreases over time in those cells. In addition to having, right, because we wash out the dox, turn off the synthesis, and watch the degradation. In addition to having the fluorescent RB, we also have a cell cycle sensor, right? So we have this Fuji cell cycle sensor, which we can use to figure out which cells are in the G1 phase of the cell cycle and which cells are in SG2 phase of the cell cycle, right? And so we can take these two things, and then we can look at the half-life of the protein in either G1 or in SG2. And so the data, these are the raw traces. And you can see that if you're in SG2, the protein is really pretty stable. On the other hand, if you're in G1, it's being degraded much more rapidly, right? And so we can take these curves, fit them to a single exponential, and then estimate the half-lives for each one of these individual cells. And the data are shown here. And you can see, for SG2 cells, protein is really quite stable. Whereas, for G1 cells, you have about an 8-hour half-life, right? So much, much less stable. And that drives, the decrease in concentration of RB during G1 phase. Okay, so protein degradation is it. What regulates that protein degradation? Well, one of the dramatic changes in the RB protein as you go from G1 to S phase is that it goes from sort of unphosphorylated or hypophosphorylated to being hyperphosphorylated, right? And so obviously that could play a role in its stability, and we wanted to investigate that. The second thing we wanted to figure out, of course, was like, molecularly, how is it sort of targeted and degraded? Why does this happen in G1 mechanistically? And so for the first part of this, I should say all of this work looking at the RB dynamics is done by a talented postdoc in the group, Shiwan Zhang, and Shiwan is really not afraid of doing work, which is helpful. We try to address these complicated problems. And so to figure out this kind of phosphorylation part of this, what Shiwan did was she generated all of these different versions of the RB protein that can be expressed from our DOX-inducible system, where we've now mutated away the various phosphorylation sites for cyclin-dependent kinase and replaced them with the phosphomimetic sites. This is like mimicking a constitutive phosphorylation on that site. And so she does this, adding more and more of these sites from either the N terminus or the C terminus, and then measures the half-life, how it changed for each one of these. And so basically what you can see is that the more and more of these phosphomimetic sites that you add, the more and more stable the protein becomes. And it doesn't really matter if you add it from the N terminus here or a C terminus here. The more sites you make phosphomimetic, the more stable the protein, really supporting this notion that phosphoregulation is an important aspect of the stability change of the G1S transition. Right. So given that, can we figure out more about the mechanism of this regulation? Okay, and so to do this, what Shiwan did was she took she constructed a couple of libraries of siRNA libraries looking at E3 ubiquitin ligases that are nuclear. And she wanted to figure out which, if any, of these nuclear ubiquitin ligases are responsible for degrading RB in G1 phase, right? But not in SG2 phase. And so to do that, we're going to use the fact that if we make a phosphomimetic RB, where all of the cyclin-dependent kinase sites have been replaced with this phosphomimetic mutations, that's stable, right? This is going to be stable in G1 phase, and SG2 phase doesn't matter. That's always going to be stable and shouldn't really be affected by the knockdown of this E3. On the other hand, if we take an RB allele and we mutate all of the phosphorylation sites to an alanine, which is unphosphorylated, right, mimics a sort of unphosphorylated version, then it should always basically be unstable, right? So we have this sort of unstable one and the stable one, and the prediction is that the knockdown of the E3 should affect the unstable one, but not the stabilized one. And so the data look like this, right? So we can basically see that the unphosphorylated, unstable RB, when we knock down UBR5, increases like quite a lot, but the phosphomimetic one doesn't increase at all, right? So this is our hit. These other these are compounds which inhibit the proteasome, and so these are sort of like positive controls for our assay. And everything else is kind of not there. So we found UBR5, added UBR5 to the second library, did the same thing again, and again found only UBR5. So this is now getting us somewhere. right? So what is UBR5? Well, UBR5 is a bona fide E3 ubiquitin ligase. It hadn't really been studied before, but then in 2023, a host of papers came out identifying various targets of this E3 ubiquitin ligase. Okay, so now we can understand what gives rise to the dynamics of the RB protein concentration. Basically, in early G1, it's unstable. And so then you go through G1S transition, it's stabilized. The amount of the protein, because it's constitutively synthesized, and then it stays roughly constant, and then it increases in SG2. This then drives this kind of like concentration dynamics where it decreases in G1 and then reaccumulates in SG2. So why would you do this? Well, in one way, we started out getting into this question because the RB concentration reflected cell volume. And so this is not the only component to why that happens, but it's a component to why that happens. And so having this regulated degradation of RB is useful because it participates in making this protein a cell size sensor in admittedly a quite convoluted way, but I guess the cell doesn't care about that. Secondly, what we and others have observed over the last few years is that, as cells become, if they're arrested in the G1 phase too long and they get too big, they start exhibiting phenotypes of senescent cells, right? So if they get too big, there's actually a threshold beyond which, no matter what the situation, the cells are unable to go on and divide again, regardless of the division signals. And so having this kind of concentration decrease may give you a way of kind of preventing that from happening, preventing you from growing too big before dividing. So, admittedly, those are two quite hand-waving things to say. So the third statement, I think we can agree on. So basically, what we have here is we have another pathway that can regulate the activity of this very key inhibitor of cell division. So one thing that is for certain is that now you have sort of two knobs that you can turn to regulate cell division. One is the kinase activity of the upstream cyclin D CDK4 complex, and then the second thing is, the concentration of the RB protein. And you might use that in the different contexts of different cell types to regulate division in different ways. Right. Finally, to sort of last thing I want to say about UBR5 relates to some work that we started doing in collaboration with Christina Curtis, who is a geneticist at the Stanford Medical School who studies breast cancer. This is a breast cancer geneticist. And so we came to her saying, we've been kind of messing around with these different data sets that are publicly available, looking at genetics of breast cancer, and we think we have a signal, but, we're terrible amateurs, and cancer genetics is its own like fully developed field. And, I'd really like to collaborate with somebody that knows what they're doing to do this. And so she introduced us to her postdoc, Lise. And so Lise has been looking at UBR5 in breast cancers and in their data. And they were quite surprised to see that it's an extremely strong signal. And in fact, if the breast cancer patient has either like an amplification, which is more than 2 extra copies, or duplication of the UBR5 allele, this is really, really bad, right? So that correlates, like if you're not going to survive, if you have that, you're going to survive a lot less than if you don't, right? So UBR5 amplification correlates with poor prognosis, which makes sense, right? Because you'd be getting rid of RB, this inhibitor of cell division. In fact, RB is not lost in many breast cancers. Only about 10% of breast cancers lose RB. However, 50% of breast cancers amplify UBR5, which degrades it. So kind of possibly the same thing. I won't go into this data, but this is looking at some cancer drugs interacting with UBR5. Right. So here we have identified really the mechanism of the second pathway into the cell cycle. The first pathway is this canonical cyclin CDK pathway, which phosphorylates RB to inactivate it. We identified sort of decreasing concentration of RB as a second important pathway. driving you into the cell cycle, and this is mediated by this UBR5 protein. And the paper is available on bioRxiv for anybody wanting more details about this. Right. So back to kind of like our main questions. We've shown you the kind of underlying mechanisms giving rise to the RB concentration dynamics. Now I'd like to talk about, our efforts in intravitreal imaging to try to connect what we're studying in vitro in human cell lines to, what happens in the context of those cells in animal. And I should say that the second piece of work is done by Mimi Shi, who's really fantastic, a postdoc in the lab, who's kind of single-handedly done everything that you're going to see from now on, which is pretty wild because it involves, mouse genetics, imaging cells with 2-photon microscopes over the course of 1 week, segmenting and tracking those cells in 3 dimensions through time, putting it all together, doing the statistical analysis. Really, it's a tour de force, single person wrecking ball. So she's great, right? So what are we doing? So and she's also a very good watercolorist. So she painted this mouse and all of the other like beautiful watercolors that you'll see that I just stole her slides. That's just how it's like that. So all credit here goes to me. So look, we have taken an anesthetized mouse, slap it under a 2-photon microscope, and then image its skin. Right? And so you can image the skin. And so at the base, you have this layer of epidermal stem cells, which are growing and dividing. And then those cells kind of differentiate and form the skin layer, die, and come off, right? So this is all in a homeostatic situation where the growth and division of the stem cells is exactly balanced by the cell death at the top. All right, so here are the stem cells, here are the differentiated cells, and then this is fluorescence imaging. So we have different fluorescent reporters that are that we can see. And so here's what a Z-stack looks like. So at the bottom here, you can see all the different stem cells. At the top, you can see these bigger differentiated cells. Okay, and so, she does this over the course of a week. Why do we do it for so long? Well, because the cell cycle time is anywhere from 2 to 5 days, right? So the compared to looking in the plastic dish, the growth is much slower. It's about 4 times slower. Not only that, the cells are about 4 times the size. In fact, some of these cells, are only a few hundred femtoliters. So this isn't all that much bigger than the biggest yeast you would look at. Right? It's actually pretty small cells. Okay, so for over 7 days, here for 8 days, every 12 hours, she takes a time point, which is a Z-stack. Right, and so then we have these sort of snapshots. We want to follow and track cells over time through the growth and division cycle. Obviously there's no software package that will do this. And so, Mimi has been implementing, various machine learning segmentation packages and stitching them together and really has put this together to make it possible to get a couple hundred cells, right? And so there's the movie, so the movie gets assembled. The cells are segmented. Each cell here is shown in a different color. And then, these are tracked over in 3 dimensions over time. So the ones that are the colors are the ones which are born, grow, and then divide over the course of the movie, right? So you can see that they're there really for kind of like, you really need the 7 days to get the full cell division cycles of the vast majority of the cells in the basal layer. Right, and so what do? So we can look here, we have a Fuchi G1 marker. So this is red in the G1 phase. So during the G1 phase, this red protein just keeps accumulating and getting brighter and brighter and brighter. Initially, when it's born, you can kind of barely see it, but then you can see it's coming up, right? It's getting brighter, brighter, and then boom, at day 2.5, the red disappears. So that's marks the G1S transition. So now we know that this cell is an S phase. it grows for a day in S phase, and then a day later it divides into two cells, into two daughter cells, right? So we can see that because we also have sort of a nuclear marker and a cortical fluorescent marker in these cells. We also have a Z-stack, so they get segmented in 3 dimensions like this, and from this we can estimate the volume of those cells, right, through time. So this is our estimate of the volume of this particular cell, so it's sort of like not growing too much, it starts growing, goes through the G1S transition, and then divides, right? And so this is, you can see, there's almost a 100-hour cell cycle. And so we can measure, the growth rate, and we're able to do this for 2 mice, about 200 cells, right? So this is the as big as our data gets, right? But it's not big, maybe a number of cells, but it's big in terabytes. Terabytes is large. But it gets distilled down to these like small little curves. Actually, we're going to use more of the terabytes later. So the first result of all this is actually that the cells growing in the animal are much better at controlling their size than what you see in the plastic dish. So in the plastic dish, this is data from Mathieu Piel's group in Paris. And what you typically see for cells growing in the plastic dish is you see that they behave a lot like what's been seen in bacteria, where you have this kind of adder phenomena where the size at birth is not, it's correlated very well with the amount of volume added per cell division cycle, right? So that, eventually you grow the same amount in each division cycle, divide by 2, and this gives you a way of like eventually slowly getting down to a target cell size over multiple cell division cycles. In contrast, what we see for the epidermal stem cells growing in the mouse skin is something much like a sizer, right? So actually there's a slope of -1. There's an inverse correlation between how big the cell is when it's born and how much it grows during the entire cell division cycle, right? So this is, this is basically the best size control that I've ever observed, that you're able to get this really good compensation. This is a kind of a perfect sizer, if you will. So it's doing a good job in controlling its size. You can ask, where in the cell cycle does that happen? We can ask if it's in G1 or if it's in the rest of the cell cycle, because we have the fluorescent marker for cell cycle phase. And in fact, it's really just in G1. And you could have guessed that before because, as you expand the cell cycle in vivo, you're going from typically 1 day in a dish to 4 days in the animal. The SG2 and M phases don't change all that much, right? You're talking about this is 12, 18 hours, something like this. Whereas the G1 phase goes from like 1 to 3 days. So it's like most of the extension in the cell division cycle takes place in G1. And that's indeed where most of the, kind of volume compensation during the growth cycle takes place too. Okay, so we can test, at least some aspects of our RB dilution model using mouse genetics. Yeah, Mimi can also do mouse genetics. So she's crossed all these mouse with different alleles and conditional mutation alleles of mice. Here, I'm going to briefly introduce this protein P107. This is very similar to protein to RB. It's in the RB family of proteins. And in mice, it's really important. And one of the reasons it's really important is because it's regulated by RB. So if you delete RB, you overexpress P107. So typically, when you only delete RB, right? So if you delete in a human, if you don't have the retinoblastoma, functional retinoblastoma protein, what do you get? You get retinoblastoma. In a mouse, you can not have the retinoblastoma protein. The mouse doesn't get retinoblastoma. To get retinoblastoma in the mouse, it also has to lose P107. And so what we're doing here is we're starting with a P107 minus background. So we've deleted P107. And then within that, we have this conditional flox allele of RB. So we can, basically add tamoxifen, and then that will kind of flip out this RB, and you generate an RB null animal. So we're comparing here RB null, P107 null to RB plus P107 null. And when you do that, when you delete both of those two things, this in red here, you lose the correlation between how long the cells are in G1 and how big the cells are when they're born, right? So basically you kill the size control by deleting these two genes in the skin. Okay, so what we saw before was that it had really good size control. And so you can plot that data in different ways. So here we're just showing for all the different cells we measure, the size of that cell and whether or not it's in G1 or whether or not it's in S phase. And you can see a very sharp kind of transition in the logistic plot as you go across the size threshold of about 550 femtoliters. And so this looks pretty good, right? There's a pretty good predictor of when the G1 S transition happens. But there's so many things going on in the tissue, right? You have like all kinds of micro, environment considerations of what's that cell is experiencing. And there's all these neighbors, there are different forces, and all of these different things could in principle be different from place to place within the tissue. And so we were wondering, what role does this play in regulating cell division? And so to do that, we thought, well, there's a bunch of different mechanisms, right? You can imagine there are diffusible ligands that would lead to local synchrony. You can imagine there'd be like contacts, shared contacts, which could also drive local synchrony, or local tissue mechanics would be specific to some particular area. And then that would also be predicted to lead to some kind of local coupling of the cell division and cell division from all of this. And so what we did is we tried to come up with all these different ways to quantify, we've tracked all of these cells through the entire division cycle. We may as well make use of all of that information. So we codified that into all of these different possible cell intrinsic features, as well as cell microenvironment and extrinsic features involving neighbors and curvature of the tissue and all of these different things. So anything we could think of, we quantified, and we put that into a logistic regression model to try to predict the G1S transition. And so to do that, well, anyway, we did that in a couple of different ways. One with the logistic model I showed you, and basically the only parameter that's at all seems to be relevant is this cell volume parameter. We can also do it with a random forest type analysis. And again, it's like cell volume is the only thing that comes out. All of these other features that we could think of putting in there don't seem to do all that much, right? Maybe a little bit, but not much. So cell size is really the major predictor, right? So it's really kind of like a cell intrinsic measurement as to when to divide. And so what kind of model does that give us, right? So microenvironment does stuff. I'm not saying that the microenvironment doesn't exist. I'm not in any way like an EGF denier, right? Like I believe in EGF, I believe in growth factors, I believe in all of these pathways. Of course they exist. I think that those pathways their activities are relatively uniform across the tissue and are linked to maintain the tissue homeostasis, right? So they're relatively uniform activities across the tissue. That then drives a certain growth rate in the cells to kind of compensate and maintain this tissue homeostasis. At the level of the stem cells, there's then some kind of critical size measurement which leads the cells to enter the cell division cycle and divide. So these are the things which are determining the cell growth. And then there's a cell intrinsic process, which is partitioning that growth into the individual cells, right? Driving you into the cell cycle. We don't see in our logistic regression models any kind of evidence of this pathway, where somehow microenvironment is like independently and importantly regulating cell cycle regression independent of cell size. So can we test that? Well, how do we perturb this and then measure this, right? And so to do that, we use our laser that we're doing the 2-photon imaging with, kind of crank it up a little bit more and like leave it focused on a single cell until that cell pops, as shown here. This is a local perturbation of the microenvironment, right? We're just popping some cell, and then we're asking, what happens to the neighbors, right? What happens to the neighbors in terms of their growth and division? And so what happens is actually they predict slightly increase their growth rate. So if you eliminate a neighbor cell, then the ones which are just adjacent to that neighbor will start growing just a little bit faster. It's not much, but it is statistically significant. However, we don't see any difference in the critical size that when they go through the G1S transition. So they increase their growth rate a little bit, but the critical size of G1S remains the same. And so that's pretty consistent with this picture, right? Where the microenvironment changes, regulate growth rate, cell growth then gets partitioned using a cell autonomous size control mechanism at G1S, driving cell cycle progression, right? So, okay, those are the conclusion to that. The major conclusions that I want you to, I guess, take away from this talk are, identified this conserved inhibitor dilution model, which is conserved from yeast to, cell cultures, human and mouse cell cultures. Important inhibitor, which is being diluted in G1 phase and by, in human cells and mouse cells is this retinoblastoma protein RB. And then that's targeted by the E3 ubiquitin ligase in some phosphorylation dependent manner. And that's like a key a building block, giving its concentration dynamics. Secondly, we asked, what's going on in a more physiological context by using this quite technical intravitally imaging. And I would say that the major conclusion for that is that there really is an absolutely, beautiful, strong size control operating in the G1S transition. And that this is a kind of cell autonomous threshold. And the important, kind of microenvironment considerations are operating at the level of regulating the cell growth rate, but not any kind of like cell extrinsic size control. All right, so with that, we'll stop and thank the group and point out Shiwen Zhang, who did the beautiful work on UBR5, and Mimi Shi here, who did the intravitally imaging work. Thank you.
Thank you, fantastic. So guys, quantitative biology can be done in eukaryotic cells.
Can be applied.
Great talk, very interesting data. The one factor I'm wondering about, what about resource allocation? In a petri dish, right? I'm assuming maybe there's sort of oversaturation of resources.
Absolutely, yeah.
In tissue, right, there's really just the blood vessels, right? Interstitial space that's sort of delivering resources to the cell. Do you think that might be maybe another sort of microenvironmental factor that
Yeah, I mean, I think the fact of like having neighbors really changes your life, right? I mean, I think that there's sort of like a lot of these cells, like the growth is not necessarily just regulated by how much nutrients can I uptake and eat, right? So like growth factors really tell the cell, like, can you even grow? Like you can have a cell sitting around in however much amino acids and whatever that it wants, and it won't do anything unless you also activate the kind of like growth factor receptor. So that's like an additional level on regulating the growth. The other one, of course, is if you have no space to grow, you also don't grow. So there's some, balance there and different pathways that are involved in kind of linking that. In terms of, applying this specific kind of like growth rate versus resource allocation that, Terry and others have been, so successfully doing in bacteria, I mean, people haven't even fully tested those type of models in yeast, much less thought of what that might mean in this like animal tissue. Like I think it's a really interesting and good question of how you do that. How does like the proteome and all of these different things change with the growth rate and the regulation? But yeah, I mean, I don't think we have a good answer to your question other than to say that it's going to be more complicated.
So in the great talk, this is a really great talk. Thanks so much. And so in the beginning, you kind of posed an interesting question of like, if you look at these different types of cells, they have different cell size.
Yeah.
And within one population, size is quite similar.
Yeah.
And so kind of going back to that, do you think is it as simple as modulating that expression of cyclin CDK to change kind of that threshold cutoff for cell size, or is the network more robust to maintaining a certain size?
Yeah, so the shorter answer is we don't know. Like we're working here in the dish. I mean, I would say that cells like inhibitors, cells like activators tend to be important like across tissues. So like RB deletions will have effects across like multiple tissues and stuff like this. So, but what sets the precise size of a particular tissue, it's not only RB, right? So like if you think about, let's think about yeast because it's easier. Like we talked about the cyclin CLIN3 and we talked about WE5. The concentration of CLIN3 was constant, but and as the cells grew in a particular condition, right? So we're not changing any condition, we're just specifying the growth condition of the yeast. Cl3 is constant and the W5 is decreasing. However, if you change the condition, right? So you change the growth rate so that the yeast is now growing slower in particular conditions, actually like the concentration of Cl3 also drops. And that has an important effect on making sure that that cell spends a longer time in the G1 phase of the cell cycle, right? So again, in the slower growth condition, as the cell increases in size, the concentration of Cl3 doesn't change. However, across conditions, the concentration of Cl3 changes and is also important in setting the cell size threshold. So it's like RB is the size dependent moving part in a given condition. However, once you start changing conditions, you can move any, one of these parts whose concentration also affects the process.
Thank you for the interesting. So, about vaccine, so you mentioned the mechanism of the concentration decrease of RB. So you said like the degradation, the phosphorylation related degradation is the main mechanism, but you also mentioned, I think somewhere before, that the dilution of the nucleoside is also cause the reason of the decrease. So which part do you think the dominant mechanism?
In this case, the dominant part is actually the degradation, because that's a time scale of about 8 hours, and the growth and dilution is a time scale of about 24 hours. So the dominant contribution to the decreasing concentration, in this case, is the degradation, rather than the growth.
Sorry to cut in here, because I had the same question, but so then how do you get this volume dependent?
It arises, like we also measure the synthesis rate,
Yeah.
right? So we measure like
It's not changing much.
protein synthesis rate, which is, it's not as exponential as yeast, right? So fighting yeast, if your cell is twice as big, you're making proteins at twice the rate, more or less. In the mammalian cell, no. Like the bigger ones are not making proportionally more cells. So you can measure the protein synthesis rate globally. We basically make a model, right? So we have a model containing like the cell growth rate, G1S transition, division, and then all of these things. You put all these things in, and then you actually do get a size dependent.
So the volume dependence is coming more.
The model produces it, right?
It's coming from synthesis.
The synthesis is also important, right? So if you just put like exponential growth, it doesn't do a good job. So you sort of also are requiring this kind of like sub-exponential growth plus this, and then you do get a size.
And the strategy is a bit different.
Strategy is a bit different. Yeah.
I had a question relating to the yeast part. So for the B5 dilution,
Mhm.
so you also need a way to like restore that high B5 concentration.
Right.
Once you divide.
Yeah.
So you showed data for the human cells where you have restoring of.
Yeah.
Do you have something similar?
Yeah. So in yeast, it is restored. So in yeast, it's like the cells are born with a fixed amount of a stable protein, which is literally diluted out by the growth of the cell. It's a simpler case. So that's easy to understand. The part about that one that's complicated to understand is how do you generate a situation where in each subdivision cycle, you make a roughly constant amount of the W5 protein, right? So that's unusual because in yeast, the bigger cells make more protein. They would make more of, typical proteins they'll make more of. So here you need a way of generating an amount of a protein per cell cycle, which is independent of the size of the cell, right? Because you want the big and the small cells to be born with the same amount of the protein. And so that is a transcriptional mechanism. So you need to basically, the yeast transcribes an amount of the W5 mRNA, which doesn't depend on size. Again, if you take like an actin or like some other housekeeping gene, you will have an amount of the mRNA which increases in direct proportion to the size of the cell. W5, the amount of mRNA is independent of the size of the cell. And so we've been working on trying to get at the mechanism that is producing that. I won't get into like the molecular aspects of it, but from a more conceptual level, as the cells get bigger, typically you scale up the synthesis rate of all kinds of different things, including mRNA. However, you don't scale up the synthesis rate of We5 mRNA. So we think of it as moving from a regime where in general, all of the genes are kind of like competing for RNA polymerase 2 to like load and initiate transcription. So the loading step, and it becomes like the limiting step. We think in the case of We5, it might not be the loading step, but rather the initiation step as limiting for initiation transcription. What would that do? Well, that would give you some kind of chromatin timescale, which would be independent of cell size, right? So the cell size, larger cells have more RNA polymerase, they're able to load more RNA polymerase more rapidly onto the genome. But then, if that doesn't help you, then you won't get a size-dependent increase in the transcription rate, whereas the We5 would stay flat. We're working on a paper on that.
But you need also something with the ribosome. So otherwise, you.
I think with the ribosome, I mean, that's kind of like part of the like global trend of making more proteins. I think the ribosome doesn't distinguish between if it's
But eventually you want to have the same level of what we find.
yeah, but the ribosomes are just treating all the mRNA equivalently. I mean, the issue is that the We5 mRNA
Okay.
amount doesn't increase in the big cells, whereas the other ones, the amount increases. Maybe that helps you.
Yeah, just a short related question. So when you divide, then you have the mother and the bud.
Yeah.
And both have probably the same amount of V5 when they divide because they're connected.
Yeah, I didn't want to get into that, but I will since you asked. Actually, you have an asymmetric division. And so even if you've made a constant amount of We5 in some cell cycle, you still have the problem of this, if that's just diffusing around the cytoplasm, you're going to get the same concentration in the big cell versus the small cell, and you don't. So there, We5 actually binds the chromatin before cell division, and so then it gets segregated with the chromosome much more equally, not totally equally, but much more equally into the mother and the daughter cells. So it gets segregated kind of proportionally to the chromosomes.
Great talk. My question is about regulation. Is there any size regulation or sensors post G1, or is that regulation gone and it's just game on until it goes through mitosis back into G1 and it has that the RB regulation again?
I mean, it depends on the organism. So in the human cells that we've studied, we don't see any evidence of like, G2M size regulation, right? Like in either the mouse or the human cells. However, I mean, there are organisms where that becomes like the dominant link of size and division. The most notable one being fission yeast. So in fission yeast, the G2M transition is what it's all about in terms of linking size and division, and does it through totally different mechanisms, still not well understood, but totally different mechanisms than what we're seeing in G1. Could it be that, other cells in the body would do it that way? It's certainly possible. I think one thing that's quite clear and has made the study of these questions pretty difficult is that there's not just one mechanism in any organism that anybody's seen. So mutation of any one of these mechanisms doesn't lead to like dramatic cell death. Maybe that makes sense. It's pretty important to link growth and division and all of these things. So it sort of makes sense that you would have other kind of like backup mechanisms, or not backup, or like other kind of overlapping mechanisms that would do this. It probably, if you think about it, we know now it's relatively trivial for a cell to generate a size-dependent change in concentration. It doesn't really cost you much to have like 2 or 3 or 4 proteins that have this kind of dependency. So why not? Like you could just add one or a little bit more of the other one and tune it and so on and so forth. And so there are multiple mechanisms. So, and we're trying to discover, what they are, especially in budding yeast. That there are mechanisms independent of We5, which are important for regulating G1S transition. We want to like understand what they are. I think like in all organisms, there are, multiple mechanisms. It wouldn't surprise me if in other human cell types in the context, they would be doing size control G2M transition rather than G1. I think we're just at the beginning of pulling apart what these molecules can do together to make this happen. Oh, a question? There is a
I was had an intuition that I've found through the talk, and you've spoken to it a little bit, but maybe you could address it directly. The density decreasing is one over volume, and it suggests that there's a constant number
yeah.
of components throughout that time period. There's a lot of competing mechanisms, synthesis and degradation, for example, that over time.
Not of We5. So We5, there's limited synthesis in G1 and is born with this constant amount. And that's why you get the inverse correlation for a We5. For the RB, it's not quite inverse volume, but then as Terry asked, what are the underlying reasons for that? And it is quite complex, involving the size dependence of the growth rate, the degradation that I've shown here.
So the number of mRNA which would transcribe these proteins depends on the cell size at birth or the cell size throughout development.
For the We5, it depends on the number of mRNAs that have existed across the, like the S and G2 phases of the preceding cell cycle. And because life isn't perfect, there's still a little bit of synthesis in G. So it's not like exactly 1 over volume, but it's within,
How much degradation?
for a physicist, it's basically 1 over volume.
So we find how much degradation?
No, in yeast, no, it has a half-life of, at least 10 hours. And you're talking about, 2-3 hour cell cycles.
So it's the burst and then they just let it.
Yeah.
Thank you.
Thank you.